Good day, everyone. If the last chapter was about building our measuring tools, then this chapter is about using those tools with the most powerful research design we have. Welcome to Chapter 13, Experimental Research. Our goal today is to understand how researchers in education can move beyond simply describing relationships to actually establishing the cause and effect. Our journey today will take us through the core purpose of experiments, the essential terminology you must know, and the major threats that can undermine an experiment's validity. We will then break down the different types of experimental designs, from the gold standard to more practical compromises, and we'll conclude by touching on the ethical responsibilities we carry as experimenters. So, what is experimental research? In one word, its purpose is causation. The goal is to determine if one variable directly causes a change in another. How is this achieved? Through a specific process. The researcher actively manipulates one variable, then observes the effect on another variable, all while controlling the environment to rule out other explanations. This controlled manipulation is what sets experiments apart and makes them the most rigorous method for establishing cause and effect. Let's simplify the core logic into four basic steps. First, we start with two or more groups that are equivalent as possible. Second, we administer a treatment, your manipulation, to one group called the experimental group, and withhold it or provide a different condition to the other, the control group. Third, you measure the outcome for both groups. Fourth, we compare the results. If the groups were truly equivalent at the start, any significant difference to the outcome can be attributed to the one thing that was different between them, the treatment. This simple logic is the foundation of all experimental design. To talk about experiments, we need a shared language. Let's cover the key terms. First, the independent variable, or IV, is the presumed cause. It is the variable that is manipulated by the researcher. For example, it could be a new teaching method, like the inquiry-based learning versus the traditional lecture. Then we have the dependent variable, or DV, is the presumed effect. It is the outcome that is measured to see if the change as a result of the manipulation. In our example, this could be a student's achievement scores on a final exam. The specific condition of the independent variable that is administered is called the treatment. Now let's talk about groups. The experimental group is one that receives the treatment or intervention you are studying. The control group is the benchmark. This group does not receive the experimental treatment. They might receive no treatment, the standard treatment, or a placebo. Their purpose is to show what could have happened without your intervention. The single most important procedure in experiment is random assignment or randomization. This means placing participants into groups purely by chance. This is our best weapon for creating equivalent groups, as it spreads out any pre-existing differences equally across groups, minimizing selection biases. The entire purpose of all this structure is to achieve control. Control is what allows us to eliminate rival explanations, all the reasons that could explain why the dependent variable changed. We achieve control in several ways. First and foremost, through random assignment. We can also hold variable constant, for instance by using only the ninth grade students in the same school. Finally, we can use statistical techniques like analysis covariance or ANCOVA to statistically control pre-existing differences like prior knowledge. Control is the shield that protects our claim of causality. With this goal of control in mind, we evaluate experiments based on two pillars of validity. Internal validity asks the fundamental question: did the treatment really make difference, or could something else be responsible? It's about the integrity of the cause and effect claim within the study itself. External validity asks, can we generalize these findings to other people, settings, or time? It's about the broader relevance of the findings. There's often a tension here. Increasing internal control, for example, by conducting the study in a stereo lab, can sometimes limit how applicable the results are to a messy real-world classroom. So what are these rival explanations that threaten eternal validity? Here are some of the common ones. History. Did a specific event occur between pre-test or post-test that affected the result? For example, a news event that relates to a topic. Or maturation. Did the participants themselves change over time simply because they grew older, tired, bored, or hungry? Or testing. Did taking the pretest itself influence scores on the post-test? This is a practice effect. Or instrumentation. Did the measuring instrument change? For example, if a grader becomes more lenient over time, or if you use a different version of the test for the post-test. Here are more critical threats to the internal validity. One is statistical regression. This is the tendency for subjects who score very high or very low in a pre-test score closer to the average on a post-test, just by chance. Or selection bias. This is a big one for class experiment. It occurs when the groups are not equivalent at the onset because participants were not randomly assigned. Or subject attrition or morality. This happens when participants drop out of the study, and if the dropouts are systematically different from those who stay, it can make the group non-equivalent. Or diffusion of treatment. This occurs when the control group finds out about the experimental treatment and imitates it, thereby blurring the line between the groups. Even if our study is perfectly controlled internally, we must ask if anyone else can use our findings. Population validity is threatened if our population is too narrow, and we generalize our study of only honor students to the entire school population. Ecological validity is threatened if the research setting is too artificial. Will a teaching method that work in a quiet, resource-rich laboratory school also work in a noisy, unfunded public school classroom? We must also consider if the results would hold if the treatment were delivered for longer duration or by a different teacher. Researchers have developed specific blueprints known as experimental designs to manage these threats. These designs are essentially plans that specify how participants are assigned. The independent variable is manipulated and the dependent variable is measured. They are primarily categorized by their ability to control internal validity threats. We have true experimental design, which offer the strongest control, quasi-experimental design, which are more practical but weaker, and the single subject design, which focus intensively on one or a few individuals. The gold standard is the true experimental design, defined by the use of random assignment. The first classic design is the pre-test post-test control group design. Here, you randomly assign participants to groups, give both a pre-test, administer the treatment to one group, and then give both a post-test. The pre-test allows you to check for and statistically control for any small initial differences. The second is a post-test only control group design. This is elegant in its simplicity. You randomly administer the treatment to one group and then give both groups a post-test. This design completely eliminates the threat of testing because there are no pre-tests to influence the outcome. However, in real-world educational settings, random assignment is often impossible. You can't randomly assign students to different schools or break up intact classrooms. In these cases, we use quasi-experimental designs. The most common is non-equivalent control group design. It looks like the pre-test post-test design, but crucially, it lacks the R to random assignment. You use pre-existing intact groups like Class A or Class B. The major threat here is selection bias. Were the two classes equivalent to begin with? The pretest is absolutely essential here to assess how similar the groups were before the treatment. Beyond these basics, there are more complex and powerful designs. Factorial designs allows you to study the effect of two or more independent variables at once. For example, you could study the effect of teaching method, class size, and achievement. The real power of this design is that it lets you see if there is an interaction effect, for instance, does the best teaching method depend on the class size or the single subject design? take a different approach, focusing intensely on one or a few individuals. They rely on taking repeatedly measures over time, establishing a stable baseline or the phase A or A phase, then introducing the treatment and seeing if the pattern changes on B phase. This is very powerful for testing intervention in special education or clinical settings. Once we have our data, how do we analyze it? The core question is always, is the difference between the group means on the post-test statistically significant or could it have happened by chance? To answer this, we use inferential statistics. A t-test is often used when we are comparing the mean score of two groups. An analysis of variance ANOVA is used when you have three or more groups to compare. And an analysis of covariance or ANCOVA is a particularly powerful tool for experiments because it allows you to statistically control a pretest score or another covariate, making your test of treatment more precise. This power to manipulate and control comes with great ethical responsibility. Informed consent is paramount. Participants must understand what they are getting into and agree to it voluntarily. We face the dilemma of equity of treatment. Is it ethical to withhold potential beneficial treatment from the control group? Often, this is addressed by offering the treatment to the control group after the study is complete. We must strive to avoid deception unless it's absolutely necessary and justifiable. And finally, the Institutional Review Board approval is not a suggestion. It is mandatory requirement for any research involving human subjects. It is a system that ensures that we do no harm. To bring it all together, the hallmark of a good experiment are clear. The goal is causality. It requires the manipulation of the independent variable and rigorous control over extraneous variables. Random assignment is the single best tool for achieving that control. A good teacher is always on the lookout for threats to internal validity, and the choice of design is always a conscientious trade-off between scientific rigor and practical reality. When done well, experimental research provides the strongest possible evidence for answering the question, what works in education? With that, that ends our topic on experimental research. I am Marlene Bong. Thank you for listening.